Welcome everyone, and thank you for joining this live webinar today. My name is Marjolein Olthoff, and I will be hosting this session. The topic of today's webinar is protecting oxygen-sensitive formulations. This webinar will review how oxygen levels in finished parenteral drug containers can be determined and controlled by using laser-based Headspace analysis. We will cover the entire product lifecycle from development to manufacturing and QC, and we will share with you real data illustrations and industry examples. Just a few instructions before we start the presentation. If you have any questions, please use the Q&A window and not the chat. I am joined by our panelist member, Josine Wilmer, Senior Study Manager at Lighthouse, and we will be available for support during this session. Additional questions after the webinar can also be emailed to experts@lighthouseinstruments.com. And of course, the webinar recording will be available for you to access or share with your team after the webinar. You will receive an email when this is online. Then I would now formally like to introduce to you our speaker, Dr. Derek Duncan. Dr. Duncan began his career at the Dutch Institute for Atomic and Molecular Physics in Amsterdam. He moved into industry holding product and application development positions, and currently at Lighthouse since 2003, Dr. Duncan is responsible for developing applications for process monitoring and finished product inspection. With that, I would like to encourage Derek to start his presentation.
Thank you, Marjolein. I understand that we have over 300 registrations from all regions of the globe, so a special thank you for those of you who are joining from inconvenient time zones, so either in Asia where it's really late at night, or perhaps the West Coast in the US where it's early in the morning. We appreciate your extra effort to join us today. As Marjolein mentioned, the topic of today's webinar will be protecting oxygen-sensitive formulations throughout the product lifecycle. And here's a little agenda of how we will do that in the next 45 to 50 minutes. We'll start with a little bit of an introduction to oxidizing formulations in general, as well as Headspace analysis as an analytical method to make a determination of oxygen levels. And then we'll go through a number of industry case studies, which are specifically meant to give you examples of how you can control Headspace oxygen in product lifecycle activities. And then we'll have a short session on some current topics, looking at some permeation versus container closure integrity. And our goal here is to end up with a few minutes at the end to collect any questions that have been submitted in the Q&A throughout the webinar, and perhaps we can pick out a few to answer here live. If we don't get to those questions, we will certainly follow up with you after the webinar. So why control Headspace oxygen levels? Here are a couple of photos of syringes, product syringes, and on the left, you can clearly see that 3 of those 6 syringes have discolored and become a yellow color. On the right-hand side, you can see that the right-hand syringe is also slightly discolored, but not quite as obvious as the ones on the left. Here are some pictures of some vials with some dry product in them. The first photo of the vials there also shows some pink discoloration due to some oxidizing reaction with the product. On the right-hand side, you see one of the vials turning yellow and looking like it has some sort of melt back, which is a combination of effects from moisture and perhaps some oxygen as well. So there is reason to pay attention to the oxygen levels in certain parenteral product containers. These formulations, and especially when you start looking into the large molecule world, protein-based formulations, you want to make sure you understand what the level of oxygen sensitivity is of these molecules, because there are some possible negative effects. We've looked at the discoloration of the product in these photos. Obviously, an interaction with the API can lead to a loss of efficacy for the product, and in the worst case, these chemical reactions or interaction with oxygen can create some toxic byproducts. So for products that are determined to be oxygen sensitive, there is a requirement to make sure you understand the product stability. And in order to do that, or to keep the product stable, you need to control the exposure of the formulation to oxygen throughout product lifecycle activities. So how do you do that, or what are some activities throughout the product lifecycle where this can become important? If you're actually in development, obviously any new formulation, you will want to determine if there is any oxygen sensitivity, and then that will allow you to make decisions about possible optimizations to the formulation. You can think about putting oxygen scavengers in the buffer, for example. And on the packaging development side, you want to make sure that you choose a package or that you choose packaging components that provide an adequate barrier to oxygen over the shelf life of the product. Once you move into manufacturing, the formulation needs to be protected during the production and filling process. So you will have some sort of process development during scale up where, for example, you will want to qualify nitrogen purging systems that are installed on filling lines, which put an overlay of nitrogen into the container before it's sealed. And then during commercial production, you might think about installing some kind of in-process control so that during filling, you actually make some measurements and monitor the performance during actual production. For quality control, obviously the stability over the whole shelf life should be verified. And it could be that there is an oxygen determination as part of the batch release. So those would be activities in QC which would require some sort of oxygen determination. Just a short word on Headspace oxygen versus dissolved oxygen. So here you see a picture of a sealed vial, and you see that there are free oxygen molecules in the headspace, as well as some oxygen that has been dissolved in the product. There will be an equilibration between these two values, and it will be dependent on the temperature, the pressure at the interface, what the dissolved substances are in the formulation. And you could decide to define stability as a function of either of these parameters. So dissolved oxygen is usually measured with some electrochemical or some optical method. Headspace oxygen, laser-based headspace is a good way to do that. You can also do it electrochemically or with gas chromatography. Um, but the traditional methods or the current methods for measuring dissolved oxygen are destructive. They can be time consuming, and as we'll see in some of these case studies, a laser-based headspace oxygen measurement is rapid and non-destructive, and this can prove to be advantageous if you want to generate some robust data in product lifecycle activities. So just one slide about the actual analytical technique. You may or may not be familiar with it already. So laser-based headspace analysis was introduced by Lighthouse into the pharmaceutical industry roughly 20 years ago. It has since been described in 2 USP chapters. And I should say that headspace analysis is a general analytical technique, and it can be used to define analytical methods in a number of different application areas. So USP 1207 is a chapter on package integrity, and laser-based headspace analysis is described as a technology for CCIT methods for determining container closure integrity. USP 922 is a new chapter that will be implemented at the beginning of next year on water activity. So headspace analysis can also be used as a water activity method, or configured as a water activity method. So what does the technique involve? We take a laser diode, which produces light at a very narrow bandwidth. That light is sent through the empty headspace of a container. You see some modulation of the light there. It's frequency modulation spectroscopy is a specific type of spectroscopy method. So we measure how much of the light is absorbed as it passes through the headspace. Uh, and because the light is exactly at a absorption wavelength of, for example, the oxygen molecule, then we know that any light that is absorbed is coming because of the presence of oxygen, and we can actually quantitatively determine the oxygen content in the headspace. As I mentioned, it is a non-destructive method, so the product is not lost. The sample can be remeasured. It's rapid, so measurements are on the order of a second, and there's no real operator expertise required. So it's a very easy method to actually use. So its flexibility, the ease of use, the robustness of the technique makes it ideal for headspace oxygen determination of sterile product throughout the product lifecycle. And we'll look at some examples of that in the upcoming slides. Um, here are some photos of actual headspace inspection platforms. We were actually part of a program called the SBIR program, where government agencies in the U.S. will provide companies with grants to commercialize promising technology. So we actually received some funding from the FDA to commercialize this technique and into these various product lines. So we have automated systems which you can use actually on packaging or production lines to do automated uh, inspection on the line, and then we have these at or offline systems, benchtop configurations, which are used in development labs or QC, and also in production for at or offline process controls. As a company, Lighthouse supplies these systems. We also offer possibilities to lease the equipment. And we also have our own analytical laboratories where we support our customers with method development and validation. So whenever talking about oxygen-sensitive formulations, I always start off with this simple case study because it really illustrates a few key principles. So what you're seeing here are headspace oxygen measurements that were made on two formulations known to be oxidizing formulations. So ascorbic acid and sodium ascorbate. And we had 3 samples of each of these formulations. They were stored at elevated temperature. We had 1 control vial, which was simply a vial with water inside. It was an air headspace, so you start off with 21% oxygen in the headspace. And what we did is over a period of 10 days, we measured each sample for headspace oxygen. So what you see here is that the ascorbic acid takes about 10 days to fully oxidize in this vial. Whereas the sodium ascorbate, after 4 days, has basically consumed all of the oxygen inside the vial. And so you see this as a demonstration of, because it's a non-destructive measurement, this laser-based headspace method enables monitoring of headspace oxygen in a single container over time. And this allows you to determine very accurately full oxygen consumption curves. So these curves here represent how quickly that amount of material or formulation will oxidize in this package. And so in this particular example, full oxygen consumption curves were required with minimum amount of samples. And also it's interesting to note here that the actual analyst time that it took to produce this data from a measurement perspective, all the measurements that are plotted here probably took less than 15 minutes. And of course, in a situation where you have scarce material, a non-destructive measurement is very advantageous in keeping the amount of material you need for the study to a minimum. So these comparative oxygen consumption studies, as for example, part of formulation optimization activities, become quite straightforward using laser-based headspace analysis. So that was a little bit of, about an oxidizing formulation. What about packaging? And here you come to the question, what is tight? Because you want to have a package which is an adequate barrier, gas barrier to oxygen in order to protect the formulation. And in reality, no containers are perfectly tight. So fused glass comes close. If you think of an ampule, for example, you will not get very much permeation through an ampule over time. But if you look at vials, it might be surprising to you to see that permeation does happen into a vial, even if the vial is sealed, stoppered, and sealed well. So here is a graph that shows the oxygen permeation into freeze-dried product vials. So this was a biopharmaceutical packaged under a slight underpressure of nitrogen. So initially there is zero oxygen inside, and then over the shelf life of 4 years, you can see that at the end of shelf life, the oxygen levels have risen to a little bit over 5% oxygen. So these freeze-dried product vials were permeating roughly 1.3% of oxygen per year. Here's some data taken for the permeation into product syringes. And here, these syringes contained a liquid product. The liquid product was known to be oxygen sensitive, so a nitrogen overlay was put into the syringes, creating a nitrogen headspace before they were stoppered with the plunger stopper. And again, we monitored the oxygen levels in these syringe samples over a shelf life. You can see here roughly 3 years, a little less than 3 years. And in this particular case, these product syringes showed roughly 3.5% of oxygen permeating per year. I should say, and we'll see examples of this later on in this presentation, it can wildly vary depending on the syringe configuration. So we saw other syringes showing greater than 10% permeation per year. And so the interesting conclusion from these simple experiments was that syringes are not as tight as vials. And so that's an interesting conclusion if you're considering what you should package your formulation in, and if your formulation is oxygen sensitive, then putting it into a syringe may mean that you have shorter shelf life. So we're going to take a short break here before we dive into the product lifecycle case studies, and we'd like to do an audience poll. So there'll be 3 questions. I'd encourage everybody to please participate. And after each question, then the results will be posted up on the screen so that everybody can see them. And I think it might be interesting results for everyone. So I believe we're going to put up the first polling question now. Okay, so the first polling question, it should be there on the right-hand side of your screen. And the question is, in what stage of the product life cycle are you currently working? Maybe it's not you're specifically working in one part of the product life cycle, but maybe the one that you identify the most with. It could be that this doesn't apply to you and you can pick others. So the options are A, development, B, manufacturing, C, quality control, D, other. So pick the one that best fits your situation and then press submit. And in 5 seconds, we'll close this polling question and show the results. Okay. Can we show the results here? So the poll has ended and we're waiting for the results. And there they are. So you can see that most people here are actually identified with the development area. So that's almost 50%, 40 something percent, 64 out of 150. Roughly a fifth of the audience is identifies with manufacturing. And then roughly 10% identifies with quality control, and then 10% others. Okay, so that gives us an idea of what kind of people might be working with auction sensitive products. Obviously, in development, uh, important to determine auction sensitivity during formulation development. So let's go to the next polling question. And this question says, which oxygen testing technique or techniques are you currently using? Uh, Perhaps you don't use it yourself, but you know that your company has certain methods that are implemented. If you know that, pick one here. You can actually pick more than one. So if you know that your company uses more than one, and the choices are A, electrochemical oxygen sensor, B, Headspace oxygen analysis, C, gas chromatography, D, other. So pick the ones that are perhaps applicable to your company, press submit, and then in 5 seconds, we will close this poll. Okay. So we're closing. The poll is ended. We're getting the results here. And there we have it. So it looks like roughly 10% of the people uh, are using electrochemical sensors. So close to 50% using Headspace oxygen analysis, gas chromatography, roughly 15%. Others also roughly 15%. Okay, so interesting. So it looks like quite a number of people are using the Headspace oxygen method. Okay. And then the last question, the third question. So the third polling question is, and this has to do perhaps it has to do with the manufacturing environment. So if you are producing an oxygen sensitive formulation, in general, what approach to monitoring oxygen during filling do you think should be done? So it doesn't necessarily mean that you are doing this, but what do you think should be done? A, no monitoring, it's not necessary. B, occasional sampling. So this would be perhaps every 20, 30 minutes pulling some samples from the line. C, statistical sampling. So sampling more often. Or D, 100% control. So the last one would be using one of these automated machines perhaps on the line to do 100% control. So pick one of those. What approach for monitoring oxygen during filling do you think should be done? And we'll look at those results. Okay, the poll is ended. We're collecting the feedback from all over the globe. And we'll see what these results are in a second. Okay. Okay, so very few say no monitoring, 3 out of 152. Occasional sampling, that's a little bit less than 10% of the respondents. Statistical sampling, roughly 1/3 of the respondents. And then 100% control, a little bit less than a third. So it looks like statistical to 100% control was chosen by most of the respondents. Okay, Very interesting. Thank you very much for participating in that poll. If you need to get rid of the polling window, there's a little arrow on the right which I think minimizes it, or the X in the window will get rid of it. And then to come back into the Q&A, if you do have a question, you can use the menus either in the bottom left-hand corner or at the top. I'm not sure how that appears in all of your computers, to get to the Q&A window to ask any questions. Okay, so let's continue with the webinar and some case studies. And I'll immediately start with quite an involved case study, and I call this stability testing or oxygen monitoring during shelf life. And there's a lot of data in this case study, so let me try and explain this well. So we were working with a manufacturer that received some complaints about some discolored product. It was liquid product in syringes. And so some feedback or complaints out of the market were that this near the end of shelf life, this product was becoming discolored, like you saw in the photos at the beginning of the presentation. So we set up a study to follow product syringes over the shelf life. And the initial levels are very low. The filling process had nitrogen purging being done to get rid of as much oxygen as possible at production. And then any residual oxygen that was left inside the syringe was consumed by the formulation. So you really saw initial levels of close to zero. Here is a graph of the results. It's a little bit of a complicated graph here, but what we are plotting are Headspace oxygen levels for 3 different sample sets defined by fill level over a 12-month period, so over the shelf life of this syringe product. So you see measurements being made on each sample from T=0 up until T=12 months. And if you zero in on samples, then you can see indeed that the initial Headspace oxygen levels were close to zero. And for certain samples, and most of the samples in fill level 1, which was a low fill level 2 was a medium fill level, and fill level 3 was a high fill level. So for the lower fill levels, you see the oxygen at some point during the shelf life starting to increase. And the question was, of course, what is going on here? Why are those oxygen levels increasing like that? Is it a container closure integrity problem, for example, or is there something else going on? If you replot this data in a slightly different way, so what we are plotting here now are the mean headspace oxygen levels of sample sets, and then as a function of the different fill levels. So you can see on the right-hand side, again, fill level 1 is the low fill level 3 is the high fill level. And again, it's very, very clear in this graph that the lower fill levels start to increase in oxygen content roughly midway through the 1-year shelf life, whereas the highest fill level actually stays quite low. What is the explanation for this? Well, this is eventually what the conclusions of this study were. Oxygen levels, according to the data, clearly begin to rise towards the second half of shelf life. What is happening is that the formulation saturates with oxygen. So oxygen is coming into the syringes by permeation. And at the moment that the formulation saturates, the API degrades, the product becomes discolored, and the consumption of oxygen by the formulation no longer keeps up with the oxygen permeation into the syringe. So the more product that you have in the syringe, the slower the rise in oxygen levels, because there's more formulation to oxidize the incoming air or the incoming oxygen. And so from the results of this study, the manufacturer had to make a decision: do we try and extend shelf life by either optimizing the formulation or by improving the packaging? Because it was clear that oxygen was permeating into these product syringes and causing this degradation. So studies like this actually got us thinking about the syringe as a package. So on the left-hand side here, you see a vial. So that's been the traditional container for parenteral products, also oxygen-sensitive products. And you think that the vial is it's a fairly simple system. You would think. You know how to cap and stopper it, cap and crimp it to get a good closure. And on the right-hand side, you see all different types of configurations for syringes. And looking at all these different types of configurations, it's a reasonable question to ask: how tight is a syringe actually? And then again, in the context of oxidizing formulations, you want to make sure that your package is an adequate barrier. So we did a little study here with two different types of syringe configurations. So very common syringe configurations on the left, Luer cone configuration, 1 ml. On the right, staked in needle, also 1 ml. And what we did is we, as part of the study, we created different subsets of each of these configurations. So here you see different subsets that we created from the Luer cone configuration. On the far left, you see a sample where we actually flame sealed the syringe at both ends, so the plunger end and the needle end. Now here, you should see no permeation into the syringe, right? It's like an ampule. And the goal of the study was to try and find out, where does the most permeation into a syringe come from? So sample number 2 in this picture has a flame sealed flange, but leaves the needle end unmodified. So if you would measure permeation into that configuration, then you would know all of the permeation is coming in through the needle end. So similarly in sample 3, here we flame sealed the needle end. So any permeation into this sample would have to come through the plunger end. And then the last one there, an unmodified syringe with permeation capable to happen at both ends of the syringe. So what we did is we took these syringes, we purged them with nitrogen, so there's no oxygen inside, and then monitored the oxygen permeation into each of these configurations. And here is a summary of the results actually, and what we are plotting here is the average permeation rate of oxygen into these different syringe configurations. So we are looking at Luer cone and staked in needle syringe, and for each of those we have these 4 subsets where we flame seal one or both of the syringe ends. And it's clear here that when you flame seal both the tip and the flange, again, that can be considered a control, no permeation into the syringe at all. If you flame seal the flange end, you get quite a lot of permeation into the Luer cone configuration, and that would be through the needle end, not so much in the staked in syringe configuration. If you flame seal the tip, then you get very little permeation into both the Luer cone and the staked in needle syringe, which means that not much permeation is coming in through the plunger end of the syringe. And then in the unmodified syringe, the results are very similar to the flame sealed flange configuration. So almost all of the permeation into a syringe in this study seems to come through the needle end of the syringe, whether it be Luer cone and staked in syringes. And the staked in syringe configuration has a lot less permeation than the Luer cone configuration in this study. So the Luer cone end of the syringe is actually the least tight location of the syringes in this study. So good to know if your formulation actually is oxygen sensitive. So for those of you who actually work in the lab, I just thought I would show you a setup that we use in our lab. So it's very helpful for development studies. It's a good setup to use for either formulation or packaging experiments. And I'll describe it here in this slide. So in this picture here, you see a glove box with an airlocked transfer chamber. And then there are cylinders of purge gas of different oxygen nitrogen gas mixtures. And of course, with this glove box, you can completely fill it with any gas mixture that you want. And then what you can do is inside this glove box, you see an electronic crimper, you see a diffusion stone or sparging stone where you can purge the oxygen out of the formulation. You see a purge gas dispensing needle for purging individual vials, and you see an oxygen sensor. And so such a setup enables the execution of quite robust science-based studies. And again, in the upcoming case studies, you'll see some examples of this. Um, where you can prepare samples inside this glove box, for example, at known oxygen levels, and then study what happens to them. And these can be finished product containers if you're looking at effects on formulation. It can also be bulk product or other materials such as excipients. If you want to study the oxygen sensitivity of any kind of material, you can decide to put it into a container in this glove box and seal it, and then study how it reacts with different levels of oxygen. This setup has also been proven to be useful for us in test method validation, because to validate an analytical method, we can produce specificity samples, so samples at known oxygen levels, and then show that the oxygen measurement or the test method is measuring all of these known oxygen levels very well. Okay, well, let's move on to a QC case study. So we've looked at a couple of development case studies. And here we had a client that was comparing their validated electrochemical analyzer, which was a destructive method, directly to Headspace analysis. And so they produced samples that were filled using different nitrogen purge rates. So they got varying levels of oxygen in the headspace. And to compare the analytical performance of both techniques, the samples were first analyzed with a laser-based headspace method. Because the method is non-destructive, multiple measurements could be performed to also determine standard deviations. And then those same samples were analyzed with the destructive electrochemical method, which gives then a true side-by-side comparison. And so just an excerpt from the results table here. You see various levels of oxygen being measured. The third column is the electrochemical measurement. The fourth column is the Lighthouse measurement with corresponding standard deviations and relative standard deviations. And you can see that the two methods agree quite well with each other. And note the last sample in this table, 13B, something went wrong during the electrochemical measurement, and because the method is destructive, then that sample was lost, and you couldn't get a value for it anymore. So, but this shows that these two methods compared quite well with each other. When we talk about doing or having a QC method, then you want to really formally validate the method. And one nice thing about the laser-based method and its non-destructiveness is that you can actually produce statistical performance reports. So what you see in the photo on the very right-hand side is a product vial. Let's say that's the vial that you want to analyze. We will actually make NIST traceable oxygen standards from the same vial that is being analyzed. So this yellow tipped container on the right-hand side is a flame-sealed empty vial, which has a certified oxygen gas mixture inside. And you can see in this table that various oxygen gas mixtures can be bought and filled into a vial and then flame sealed shut. So here you have standards that are at 0, 1, 2, 4, 8, and 20% oxygen. And you can measure these standards 100 times, and you can very easily get error standard deviations from these results. And then you can really validate this as an analytical method using various approaches for validation and determine accuracy, precision, specificity, etc. So you have full insight into the performance of your method. And in these systems, you can also save all your data in a compliant database with a full audit trail. Again, from a QC perspective, very, very important. Here's just another QC example, and this really came from a formal PQ. Again, a side-by-side comparison with a validated system. Again, it was electrochemical. And by doing this correlation study, then to an existing validated method, this enabled a straightforward switch to a new, better method for doing oxygen determination. A similar case study, but now with gas chromatography. And you see here some example tables of side-by-side comparisons using gas chromatography and headspace analysis. Again, quite good agreement between these two methods. So you could, again, do a direct correlation study and compare to your validated GC method. What was interesting in this case is that the client actually did a business case looking at, for example, analysis times of the two methods. Is it destructive, yes or no? Do I have the capability to put it at line for monitoring during production? What are disposal costs? And then they came up with a cost per test and determined that they could easily replace their existing gas chromatography with this with the Headspace analysis method. And they were also looking into considering an inline system for doing 100% monitoring during filling. Okay, so now let's go to some manufacturing case studies. So if you're working in manufacturing and having to fill oxygen sensitive product, how can you use this technique to improve or add value to your activities? So a lot of filling lines will purge the headspace if you have an oxygen sensitive formulation with an inert gas. Most of the time it's nitrogen. Sometimes you see argon being used. And a nitrogen purging process can typically achieve 2 to 5% of oxygen in the headspace of vials. And obviously a good method for measuring or monitoring can help you qualify or monitor these processes. And here's an example of some data where a filling line was being used with a nitrogen purging system, and you see various flow rates on this graph. So if the nitrogen purging system was off, so the flow rate is 0, then you see that all the vials produced on the line have atmospheric levels of oxygen, 21%. You turn on the nitrogen purge, and all of a sudden the values of the headspace oxygen goes down to between 2 and 5. And then at varying levels or varying purge rates, you can see the corresponding oxygen values. And if you look here, this data was being collected at a line speed of 90 vials per minute. So you can really monitor at a high speed. I mean, this is actually a relatively low production speed, but machines can be configured to monitor at hundreds of vials per minute. And there is a trend of more filling lines being implemented with nitrogen purging capability. And there seems to be an increasing trend then of nitrogen overlaid products in the parenteral space. And there's correspondingly then a need to monitor this process. So here's an example of qualifying a filling line that has a nitrogen purging system on it. This was actually a syringe filling line. It was a brand new syringe filling line. And for the first time, this line was going to be used to fill oxygen sensitive product. So there was a purging system installed on the line. And then they started to run syringes during the setup, and we're measuring what the oxygen levels were as the purging system, as different parameters on the purging system were used. And you can see here, series 1, the target was to actually reach 5% oxygen in the headspace of these syringes, but clearly there are some purging needles here that are not consistent or not purging that efficiently. You see some outliers here up to 14, 15%. This data was collected very easily with an at-line benchtop analyzer. The operators, it's very easy to use, so operators could just take syringes off the line as they're being produced and make a measurement. And so very quickly they were able to adjust the filling line parameters, the nitrogen purging system parameters, to give them the optimum target level of under 5%. And so you can see in series 2, after some optimization, every purging needle was performing adequately. So this at or inline implementation of Headspace oxygen monitoring, enables, number one, efficient process optimization and qualification. And then during the actual commercial production, you can use this for some IPC monitoring. One last manufacturing case study. This was a case where a client was using an older filling line and had to fill a product that needed this nitrogen overlay. And you can see here that there was a dead space between the what's called the post purge in the filling to the stopping station. And the open vials had some resonance time in this space. And so here, because nitrogen is actually lighter than air, you would see some nitrogen escape out of these vials before they were actually stoppered. So there was a requirement to get down to 2%, which they couldn't do. And so the advice from the filling line supplier was to install a nitrogen overlay system above this dead space. So you could call it a nitrogen tunnel. But when they tested this, it actually made the situation worse because this tunnel caused turbulent flow around those vials, which actually sucked in air from around the filling line. And actually you saw higher levels of headspace oxygen in the vials with the tunnel compared to without the tunnel. So finally, the solution was an extended post-purge arm for the post-purge needles, and this minimized the dead time and thus the dead space, and therefore the residence time before the stopping station. And you will see most filling lines these days have learned this lesson, and so the stopping station now is as close as possible to this post purge on the filling line. All of this work was done with a benchtop Headspace analyzer right at the line, so it was very efficient to collect all the data for both the optimizing and for the validation. There was a significant reduction in production time because the line didn't have to be shut down for a whole bunch of studies, a long period of studies. The data was generated quite quickly. The filling line could be validated for optimal purge performance, resulting in 2% headspace oxygen. And they could even validate a software solution to reject a fixed number of vials after a machine stoppage. So when the filling line stops, then a large number of vials had to be rejected because all the nitrogen would escape, and they were able to actually validate a software solution for that. Okay, so those are a few case studies in the different product lifecycle phases. We're going to move now into the last section of this webinar where we've picked the topic of permeation versus container closure integrity testing. So we're always looking for interesting data, which is current, to present as case studies. So here you see some data from a recent publication in the PDA journal from West. And in this particular study, various vial stopper combinations were used, a lot of different combinations. They were stoppered, capped and crimped, so they were sealed. Helium leak testing confirmed that CCI was good. And then these vials, these empty vials were followed over a 12-month period. Um, the stoppers were different stoppers of different rubber formulations, so you can see the different stopper groups in this graph, and the different capping and crimping settings were used to achieve different compressions of the stoppers. Um, and what you see here is these vials had an air headspace. They were stored for 12 months in a nitrogen environment, and you can see that oxygen is permeating out of these vials into the nitrogen environment. So despite the fact that they are sealed well, and helium leak confirms this, you do see, you still see gas exchange between the inside and the outside of the vial. And this, of course, agrees or confirms the results that we showed at the beginning of this webinar, where we tracked the oxygen permeation into product vials, freeze-dried product vials over the shelf life. And if you look at the numbers here on the graph at the left, it is roughly between 0.7 or 1.5 percent of oxygen over a year, matching roughly what was seen in these product vials on the right. And the permeation depends most strongly on the rubber formulation, so there's differences there, but it does not seem to depend much on the compression level, which really indicates that the permeation is happening through the actual rubber material of the rubber stopper. Here's some more recent results looking at oxygen permeation. This is a presentation that was given at last year's Cell and Gene Therapy Conference at the PDA. Again, this was also research done at West. And here, what they looked at was permeation of oxygen into a COP vial as a function of temperature. So now you're talking about a polymer vial, COP, it's not glass. So you will want to know what are the permeation properties of this vial, especially if you are thinking about putting oxygen sensitive product into it. And on the left hand side there, you see permeation into two different size COP vials, and this is over a period of a little bit less than a year. And you see that over the course of that year, you're seeing oxygen permeate into this container. On the right hand side, you see the permeation as a function of temperature. So now they have some room temperature samples, refrigerated minus 20 and minus 80, and you can see that the minus 80 has the least permeation, which is what you would expect, and in fact it's close to zero. So if you zoom in on that and look at the permeation into the COP vial at minus 80 and compare it to glass, the data actually indicates that the permeation of oxygen through the COP vial during deep cold storage is actually quite similar to a glass vial. And especially the cell and gene therapy people would find this interesting, because a lot of the cell and gene therapy formulations actually require this deep cold storage at minus 80, even cryo. And this data shows that the permeation rates at these temperatures are actually quite low and close to zero. So those were some interesting recent results. Here, just some results out of our own lab, again, showing that you can actually track oxygen permeation dynamics during temperature excursions. So we had different samples here that were stoppered vials with nitrogen inside. So initial levels of oxygen, which is what we're plotting here, are zero. And then you see and these were also a polymer container, by the way. And so you see some quite rapid permeation into this polymer container. But when the samples are temporarily taken out of room temperature and put into cold storage, and again here you see refrigerated minus 20, minus 80, you see that these temperatures basically cut off the permeation. They're stored during cold storage for a little while. They're brought back out to room temperature, and then you see the permeation pick up again, and everything eventually converges to the same endpoint. But it's interesting to see these permeation dynamics during these temperature excursions. A last case study here, oxygen permeation as a function of rubber formulation. So here you see the different stoppers, different formulation stoppers. So we mentioned in that initial West publication, seeing permeation vary according to the stopper formulation. Very clearly here, you see that it does, the permeation does indeed depend on the particular stopper formulation. And the relative dependence on temperature also depends on what the formulation is. So here you see different rubber formulations that were used are stored at room temperature and 40 degrees. Again, the higher the temperature, the faster the permeation. And there's variability between the different rubber formulation groups. So again, important to know in packaging development when you're trying to choose your components, and it's not necessarily, there are many reasons for different stopper formulations, whether that's extractable and leachable profiles or interaction with the formulation or permeation. So all of that data needs to be collected so that an informed decision can be made. You might ask the question, is that are those elevated oxygen levels really due to permeation, or can it be due to a container closure integrity failure? So what we're showing here is the same data from the last slide, but now plotted on a full oxygen scale. So now from 0 to 25%, so that you can also include oxygen, atmospheric levels of oxygen. And now this graph here on the right shows oxygen ingress into a container, a vial, which has a real defect, so a known defect, a laser drilled hole of different sizes here, so 4 micron hole, a 9 micron hole, and a 16 micron hole. And you can see that the oxygen in these CCI defective containers rises very quickly. So now you see a time scale of minutes for the graph on the right compared to a time scale of days on the graph on the left. So the permeation rates through the stopper are orders of magnitude smaller than the natural diffusion rate through a CCI defect in a stoppered vial. And again, there's a reference to a recent publication there. Again, a similar graph, but now on the same plot. Going back to those syringe studies, we did compare the permeation rates of the syringes to oxygen ingress through an actual CCI defect, this time a 2 micron defect laser drilled into the syringe barrel. And again, here you see that a CCI defect will cause oxygen to rise in orders of magnitude faster than the permeation. This is actually an example of inherent package integrity determination. If you're familiar with the USP 1207 chapter, the concept of maximum allowable leakage limit, if you're able to produce data like this, then you can make very informed decisions about packaging for your oxygen sensitive formulations. You know, how much am I allowed to leak in these packages? And a leak does not necessarily mean a CCI failure, but could also mean a permeation rate. Okay, and the last slide, I'm showing here a photo of what we call a CCIT vessel for headspace gas ingress testing. These are vessels that can be evacuated and overpressured with any kind of gas that you want. We've been using them primarily in combination with headspace analysis to do CCI testing, but you can also use these vessels for permeation studies. So you can imagine putting containers into these vessels, filling them with a certain gas, sealing it, and leaving them there for months or a year and measuring permeation rates. If you use this vessel for CCI testing, and this is a whole other topic, we also have another webinar on this that you can watch. but you can develop CCIT methods based on Headspace gas ingress, which are very similar to a blue dye test, actually. So instead of using a blue dye bath as a tracer, you're using a gas bath as a tracer. And when you store samples inside the CCIT vessel, you will get ingress of the tracer gas, which you can then measure with Headspace analysis. Okay, so let's come to our summary. An increasing amount of product needs an inert gas headspace and optimal primary packaging to protect the formulation from oxygen. If you have a rapid non-destructive headspace oxygen analysis method, this enables you to develop methodologies that give deep insight into product and process. And finally, generating robust science-based data really enables data-driven decisions about how oxygen-sensitive product needs to be formulated, packaged, and manufactured. And this is what the regulator wants, actually. They want you to be to know your product and to know your process. So the main message here is not necessarily all the details of this data. I hope that didn't overwhelm you, but the fact that you can generate this type of data, which then enables you to make good decisions about product and process. Okay, I see that we're almost out of time. In fact, we're at 5:30. So we won't have as much time as I would have wanted to cover questions. So let me pick out really quickly one or two questions here. And again, if we don't get to your question, you will get a response by email. So one question, can measurements be made for HDPE bottles? So here we're talking about plastic bottles. The answer to that is maybe, if the bottle is translucent or transparent to laser light. And a good test is to take your laser pointer, go into a dark room and see if the laser pointer light goes through the container. If that's the case, then you can make a measurement on it. So that's a good little test. Let me see another quick question here. Could a headspace measurement be affected when using molded vials versus tubular vials? That's a good question. Molded vials are very bad glass quality. And again, the main question here is, can you get laser light through the molded vial? And the answer to that is almost always. We work a lot with molded vials. And so in general, that is not a problem. Also, amber colored vials could be measured. We've seen a few very dark amber molded vials, which then become difficult. But again, the key is getting some laser light transmission through the container. Okay, I'm afraid I don't have time to go over the other questions that have come in. Again, we will have our application experts review all the questions, and you will receive some feedback via email. You can also contact us directly with your question. Like to again thank everybody for joining us. We hope that this information was useful for you, and we look forward to seeing you on future webinars. Thanks again.